All right, happy Wednesday. Everybody's energetic. It's nice outside. The plants are blooming and spewing their pollen. So, judging from the amount of noise in the room, I suspect that a lot of you probably weren't doing this, which is totally fine. We can do it right now all together. It won't take very long. If you start out, we're going to use this sort of framework, this question, for most of the class today, especially, you know this if you've looked through the slides. This figure shows up a lot. You start out with a pure breeding parent that's got purple flowers and long pollen, so one combination of phenotypes. It's big P over big P, big L over big L. So previously we had already rewritten those in terms of genotypes, that nomenclature that we normally use. So if you cross a purple long pollen flower and a red round pollen flower and make F1s and you self the F1s, you don't have to draw out the Punnett square. How many different combinations of phenotypes could we have in the F2 generation? What are all the possible combinations of flower color and pollen size? You've got two color, and you've got two different pollen types. So hypothetically, what are all the combinations of these traits that you could make? Right, so you could either combine purple with long pollen or purple flowers with round pollen. And you could combine red flowers with long pollen or red flowers with round pollen. So there are 4 different phenotype combinations that you could make. So one dihybrid cross, right, each of these individuals, the F1s are dihybrids. Big P over little p, big L over little l. So when you cross a dihybrid to itself or to its sibling, you should expect to find the 4 different combinations of traits. So what are the haplotypes that this F1 individual produces? So big P over little p, semicolon, big L over little l. So 2 genes, one that controls flower color, one that controls pollen shape. They're on separate chromosomes, so they should independently assort. How many gamete types get produced by this individual flower? So independent assortment tells us that at the first locus, at the P gene, there are 2 alleles. How many alleles at the second gene? 2. There's a big L and there's a little l. So how many different possible combinations of P's and L's can you make? Should sound familiar. It's 4. You can have big P's with big L's, big P's with little l's, little p's with little l's, little p's with big L's. Those are the 4 different gamete haplotypes that are produced. And that's what we've been talking in class, the last couple of class sessions. So you can get big P's with big L's, big P's with little l's, little p's, big L's, little p's, little l's. So an individual dihybrid will make 4 types of gametes, 4 haplotypes, and if you cross 2 dihybrids together, or self one, you expect 4 different phenotypes, the 4 different combinations of the traits. So there's a lot of 4 going on here. 4 different phenotypes produced by this F1 plant in its offspring. It makes 4 different haplotypes of gametes. Any questions at this point? Most of the review. At this point, we could set up a Punnett square. So you could put the haplotypes of the gametes across 4 columns and down 4 rows and figure out the genotype of the next generation. And that's what I've done for us so that we don't have to spend a lot of time drawing stuff. That's exactly what this is. So the F1 haplotypes, the gamete genotypes, across and down. And then we don't have to fill in the square. There are all of the possible genotypes that could be produced in the next generation in the middle of the Punnett square. Bless you. How many of each genotype, how many genotypes are there, first of all, and how many of each are there? 16 total squares in here. So the first thing we should do is just look at the patterns. First, we want to talk about genotype frequencies. So how many out of the 16 are pure breeding, let's start with? How many of these 16 squares represent pure breeding F2 genotypes? These are the F2s. Okay, I counted, okay, so I heard a 4. Did anybody else agree, disagree? Care to disagree? How many pure breeding genotypes? Where are they found in this square?
Diagonally down from the top left.
Diagonally down from the top left to the bottom right. So how many big P over big P, semicolon, big L over big L genotypes are there in this table? That one. So there's 1 out of 16 individuals that we expect to find that's homozygous at both loci, both genes, for the dominant allele. How many that have the same except recessive? The little p over little p, little l over little l. It's just that one. So we're starting to prepare the ratio of genotypes. So if you think bigger than Punnett square, this just represents the 16 types of haplotypes that produce. But of course, a plant doesn't just have 16 offspring. It could have hundreds of thousands of offspring. So you have to think bigger. How many individuals in the whole population, and what's the ratio of the different types? So all 4 of those that are circled there on that diagonal only show up once in the table, in the Punnett square. So we know that there are going to be 4 genotypes, at least, maybe more, where they're just represented once in the table. Do you see any genotypes where there are more than one? Where are they? So for example, here's big P over big P, big L over little l. There's another one right there. There are 2 of those on the table. And the same is true for that genotype. So the 2 I've circled in red, those are paired. So in the 16 squares of the Punnett square, each of those 2 show up twice. And the same is true down here. And right there. So there are 2 big P over little p, little l over little l genotypes in the 16. Okay. What have I left off?
4 heterozygotes.
The 4 heterozygotes. Now I better choose a different color. These 4. So there are 4, what do we call these genotypes? They're heterozygous, 2 genes. These are, what genotype do these, this genotype's already drawn on this diagram on the right. Which individual is it? Big P over little p, big L over little l. Those look like the F1 dihybrid. So there are 4 dihybrid genotypes. So there's the genotype ratio. Every time you do a dihybrid Punnett square, don't memorize this. I'm just trying to help you see patterns. It may be helpful at some point. We've accounted for every one of those 16 different genotypes in the square. There are 4 genotypes that only show up once. Those are the ones in this ratio down here. There are 4 genotypes that show up twice in the table, and there's one genotype, the dihybrid genotype, that shows up 4 times this diagonal. So the genotype ratio you get, you do not have to memorize the 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio. So every time you cross a dihybrid with itself, or you self a dihybrid, you expect to see 9 different genotypes. And they're in these relative frequencies. That's what the 1, 2s, and 4s represent. So 1/16, for example, has that little p over little p, little l over little l genotype. So you expect 1 out of every 16 F2 individuals to have that genotype. You expect a quarter, 4/16, to be dihybrids, and so forth. So that's genotype ratio. Yeah?
Do you have to write that ratio in that order?
You don't have to.
Okay.
It's just easiest for me to remember because it's symmetrical. I suppose you could call it a 1 to 1 to 1 to 1 to 2 to 2 to 2 to 2 to 4 ratio. That's fine. And I will never ask you to repeat the ratio. Like, that's why I said don't memorize it. But I want to get you thinking about getting a sense for the combinations of alleles that are produced by crossing a dihybrid. You get 9 different genotypes. But there are some that are more rare, and there are some that are more common. So the most common genotype you get out of selfing a dihybrid is the dihybrid again. The big P over little p, big L over little l genotype. So how is this useful? Well, it kind of gets back to that original question. If we're trying to make a particular trait and we want to do it as efficiently as possible, or if we want to generate the most individuals that have a particular type of trait or combination of traits, this ratio is really useful because you can use it to figure out how frequent is that trait going to be. Now let's switch. That was genotypes. That was looking at what are the alleles inside the plants. Now let's talk about phenotypes. A couple of classes ago, I started you hopefully thinking about, with knowledge of dominance and the knowledge of genotype, you can predict the phenotype of an individual. So if I tell you they're heterozygous, you could tell me, well, let's see, if you're heterozygous, you have one dominant allele and one recessive allele, what's your trait going to be? The dominant trait or the recessive trait? So you're heterozygous, you're going to show the dominant trait. So how many different phenotypes are in this table? Ignore the genotypes. Well, we're not going to talk about DNA anymore. We're going to talk about traits. Red, purple, round pollen, long pollen. So 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 is the genotype ratio. What's the phenotype ratio? How many of these plants? Is that purple? Yeah, close enough. Says the colorblind male. How many of these plants are going to be purple flowered in the F2, inside the Punnett square? What's the genotype we look for to tell which plant is going to be purple? It's either going to be big P over big P, or could be heterozygous. Either of those would be purple because purple's dominant. So we can make a circle around every individual that's got at least one big P, 1 or 2. So what are the relative numbers of purple plants to red plants in the F2 generation?
12 to 4.
Yeah, so it's 12 to 4, which we could reduce to a 3 to 1 ratio. So out of every 4 plants you look at, about 3 of them should be purple. Here are some red plants. The quarter of the F2 offspring that don't have any big P alleles. So now we're talking about phenotype ratios. not genotype. So we've converted genotype to phenotype, and now we're counting up how many plants have certain numbers of traits. 3 to 1, purple to red. What about long pollen to round pollen? Oh goodness, so it's going to be, what's the ratio going to be?
The same.
It's also going to be the same, it's going to be 3 to 1. Because each of these genes acts as though you're making a monohybrid cross. You're selfing a heterozygote. You get a 3 to 1 phenotype ratio from a monohybrid cross. Let's circle them all. So everybody that's got a big L, although this is going to get kind of funny looking because I have to go in and out and around the tree. And then there's one individual right there that doesn't have any big L's. So all of this space in here are the individuals that have big L's. So there's one square there that doesn't have any big L's. There's one there, there's one there, and there's one there. So 12 out of 16, big L. 4 out of 16, no big L's. So it's, again, that 3 to 1 ratio. Expect about 12 long pollen plants to every 4 short round pollen plants. But we're talking about 2 different traits. So what have we not done yet? What's the point of doing a dihybrid cross? We're trying to combine 2 types of traits together. So now, red. 3 purple to every 1 red. So how many purple long and how many purple short? And how many red long pollen and how many red round pollen short round? What's the relative frequencies of those? You predicted on the first slide there were going to be 4 phenotypes out of this cross. All 4 combinations of the 2 flower colors and the 2 pollen shapes. So what are the relative ratios of those 4? We've got purple long. We've got purple round, we've got red long pollen, and we've got red round pollen. So how many squares are circled both in blue and black up there? Those are the individuals that have both the purple flower colors and the long pollen. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Am I doing this right? What am I missing? 7, 8, 9. So there's the 9. All of those individuals have a big P and a big L. So those will be purple long pollened plants. Okay. And then there are 3 purple with round pollen, there are 3 red with long pollen, and then there's only one individual, the one down here in the lonely bottom right corner, that has red flowers and round pollen. This is what happens when independent assortment is happening. This is when Mendel's second law is working. You expect to see a 9 to 3 to 3 to 1 phenotype ratio. And that's if you're mathematically inclined, which I'm not necessarily. That's what happens when you multiply 2 3 to 1 phenotype ratios together. So you could think about drawing a Punnett square of phenotypes almost, where a quarter of the flowers are purple, or 3/4 are purple, a quarter are red. 3/4 have long pollen, a quarter, the relative heights of the table, have round pollen. So you get all the random combination of those 2 traits. Relative ratio of 9 to 3 to 3 to 1. talked about genotype ratio, phenotype ratio. The goal is to have everybody comfortable converting genotypes to phenotypes and being able to predict how many of each type of genotype and phenotype would be produced by a cross. Not necessarily just a dihybrid cross like this one, the 16 square Punnett square, but maybe something more simple. Do you have any questions or concerns at this point? All right, so here's where we get into things that are a little bit kooky, weird, strange. Every time Mendel, have we talked about Mendel yet? Other than his laws, did I talk about P's at all? Good. Because you've already heard enough about Mendel's P's, except for today. So Mendel worked with a number of different traits. He crossed a lot of pure breeding parents together to make dihybrid F1s. In this is one example, right? P shape, round versus wrinkled, P color, yellow versus green. But he worked with a lot of different traits besides these two. And every time he set up a cross, or at least that's what he said happened, he saw a 9:3 to 3:1 ratio in the F2 generation. So there was always one phenotype in the F2 generation that about 9/16 of the individuals had. There were two that were 3 out of 16, and there was one phenotype that he rarely saw, 1 in 16 times. And it wasn't just for this combination of traits. It was flower position and whether or not the pea pods were, smooth or wrinkled or something like that. So that's where we start moving from independent assortment, Mendel's second law, into what happens when Mendel's second law doesn't work. So this is where we learn something new about genetics and about biology, is when Bateson and Punnett, two geneticists that came later than Mendel, came along and did some similar crosses of plants and found something that didn't agree with what Mendel found. And that's when we find really cool things out about how biology works, is when somebody comes along and finds an exception. Like, hey, Mendel, your second law is not working. Why is it? So that's when we start talking about linkage between genes and comparing it to independent assortment. So this is Punnett of Punnett square fame versus Mendel. So Mendel came up with the law of independent assortment. Punnett came along later and did an experiment to prove that Mendel's second law does not always happen. There are exceptions. Linkage is the exception. So that's what we're here to talk about. So I think we're used to thinking about nomenclature of genotypes in this way. If we have a dihybrid genotype, and we're talking about independent assortment, we might draw that to represent each vertical stick representing a chromosome. So we've got a diploid individual that's heterozygous at 2 loci. And those two genes, A and B, are on 2 different chromosomes. Okay? And we write that, as you know by now, like this. Linkage, what's the concept of linkage? How am I going to redraw these 2 genes as linked genes? This is where Mendel's rule gets broken. These are linked genes. How do you describe that? How does it differ from what I drew up top? The 2 genes,
They're on the same chromosome.
they're on the same chromosome. So this gets to an important point. When genes are on different chromosomes, we call them unlinked because physically they are unattached. They're not linked. But when 2 genes are found on the same chromosome, then we call them linked genes. And we write the genotypes, I apologize, bless you. Unfortunately, we have new nomenclature for linked genes. So you know that the slash separates alleles that are on the same chromosome. So we've got two different chromosomes. So there's going to be a slash that separates what's on one piece of DNA and what's on another piece of DNA. Because we're talking about the same chromosomes, the nomenclature for the genotype of that individual would be big A, big B, no semicolons, nothing, just big A, big B, because they're linked. That's how we know they're linked. There's nothing in between the A and the B. They're on one piece of DNA. The slash means now we're going to start talking about a separate chromosome, and that's where the little a and the little b are. What would the genotype be in this case? We've got two pieces of DNA, so two linked genes, A and B. What do I write on the left side of the slash? Big A, small b, and then over little a, big B. Are the genotypes fundamentally different? What one word would you use to describe either of those genotypes? This one or that one? Heterozygous at how many genes? A gene, B gene. So what do we normally call a heterozygote at two genes? A dihybrid. These are both the same these both have the same genotype. They're both big A over little a, big B over little b. So these two genotypes are equivalent. That is, genetically, these individuals are identical. The only thing we're adding now here is information about which allele goes together on the same piece of DNA with which allele. In the case of the left, the big A and the big B and the little a and the little b are together. On the right, it's one capital and one lowercase letter. So exactly the same genotype, but now we're just looking at the linkage between alleles. Lastly, for this concept anyway, just like in this case, a little b by itself could be the haplotype of a gamete. Whatever's written on one side of the slash is what will go into a gamete. We would call that a haplotype. The same is true for talking about two linked genes. So when that diploid cell undergoes meiosis, one of the products of meiosis would have a little a, big B haplotype. So one of the gametes will have that combination of alleles. And that's only because they come together on the same piece of DNA. So they segregate together. Those two alleles of two different genes, the little a and the big B, don't segregate independently. That's Mendel's second law, because they're linked. They're physically stuck together. So at meiosis, the little a and the big B don't always, although we'll talk about recombination, the exception to the exception, later. This is why Mendel's second law doesn't work all the time when two genes are found on the same piece of DNA. At meiosis, the little a and the big B go together into a gamete. So this is just nomenclature. Linked genes, you write all of the alleles that are on one piece of DNA next to each other on either side, on both sides of the slash. Okay, so time to put this to use. Please draw what the gametes would look like, both from the P0 and the F1 generations. So this is assuming linkage. So the parent on the left, the P0 on the left, has two big P's and two big L's, and the parent up here has two little P's and two little L's. So if you fill in, what do their gametes look like? What are the haplotypes that go inside those circles? What's true about pure breeding genotypes and the gametes that they produce? If an individual is homozygous at every locus, how many types of gametes will it produce? How many haplotypes? How many different haplotypes? Does every gamete have a big P, or are there some that have big P's and some that have little p's in this case? Right, so I'll guarantee all the gametes are going to get a big P. What about the L? The same. So that's the haplotype of every single gamete from the parent in the left side. And the same is true for the parent on the right side. By virtue of being homozygous, it doesn't matter if the genes are linked or not, it's homozygous. So it can only produce gametes that have one haplotype. Parent on the left, big P, big L. Parent on the right, little p, little l. So when those come together in the F1, that F1 individual has chromosomes that look like this. It got its big P, big L chromosome from the parent on the left, and it's got its little p, little l chromosome from the parent on the right. So it's a dihybrid, big P over little p, big L over little l. It's just that in this case we have a little bit more information. We know that the two capital letters are on one chromosome, and the two lowercase letters are on the sister chromosome, the homolog. So when that cell, F1 diploid cell, does meiosis, what are its gametes going to look like? What types of haplotypes does it produce, and how many of each? It's exactly the same as the parents. So these two alleles go together. Those two alleles stay together. So you get a 1-to-1 ratio. You know out of every meiosis you make 4 gametes, but they're in a 1-to-1 ratio. So you get half 2, that is, big P, big L gametes and 2 little p, little l gametes. When genes are linked, when they travel together because they're stuck together on the same piece of DNA. The absence of independent assortment, because independent assortment is talking about genes that are on 2 different chromosomes, and it's the random alignment of chromosomes during meiosis, as we've discussed before, that's the cause of independent assortment. When genes are stuck together, they can't independently assort. They dependently assort. Yeah?
So if they weren't linked, then would you have 4 different haplotypes?
This. You just make 2 types of gametes. You never get any big P's with little l's. You never get any little p's with big L's. That's correct. So if they weren't linked, if we drew this the way we would and have in previous classes with the 2 genes on different chromosomes, then we're back at the situation where after synthesis, you get the 2 capital P's and the 2 lowercase p's next to each other, and the question is, this is the independent assortment part. Because those genes are on separate chromosomes, the chromosomes can line up in one of 2 ways before the first division. You can either get the big P's and the big L's on the same side, dividing into daughter cells, with the little p's and the little l's going their own way. And at the same time, you also get the big P's and the little p's lined up where the capitals don't go with capital letters and lowercase don't go with lowercase. So when those go through the first division of meiosis, these are separate cells going through meiosis in the same individual. You produce an equal number of big P's with little l's and little p's with big L's. So you get the 1 to 1 to 1 to 1 ratio of 4 different gametypes. So this is the essence of the difference between independent assortment. So bottom line, summary. Genes are on 2 separate chromosomes, independent assortment happens. You make 4 different types of gametes from a dihybrid. These are the 4. When those genes are stuck on the same chromosome, what we've been talking about up to this point happens. So with that information, we can make a really easy Punnett square. If P and L are linked, this F1 individual, their diploid cell looks like this. Heterozygous at 2 loci, a big P and a little p, a big L and a little l. You just told me the gametes, the previous slide. You told me which gametes this individual produces. It's either going to be big P, big L, or it's going to be little p, little l. And if you self that individual, right, like crossing it to itself, that individual only makes 2 types of gametes. Big P with big L, little p with little l. It's way easier than a 4x4 Punnett square. This is what linkage does. What's the effect of linkage? How does linkage impact the outcome of a dihybrid selfing? The first square we did today, 16 square Punnett square, 4x4, we talked about genotypes and phenotype ratios, 9 to 3 to 3 to 1 and all that. There was a lot of variety. There were 9 different genotypes produced and 4 different phenotypes produced. How many genotypes and phenotypes do we get when the genes, same traits, but if they're linked? If those genes just happen to be on the same chromosome, how does that affect the number of genotypes and phenotypes? So if you fill this in, how many different phenotypes are there?
2.
So we've got plants that have at least 1 big P and at least 1 big L. So all of those 3 out of 4 plants, what's their phenotype?
Purple flowers with long pollen.
Purple long. So that looks like parent 1. What's the other phenotype? Looks like parent 2. Red flowers, brown pollen. Totally different than what we saw when genes were independently assorting. Independent assortment gives us all the variety that we saw, that is, the 2 missing phenotypes that we saw with independent assortment that we don't see in this cross. Okay. Both examples, taking an F1 dihybrid and selfing it. But when the genes are unlinked, when they're on different chromosomes, we see additional phenotypes, purple flowers with round pollen, the traits that are not seen in the parents. Purple flowers with round pollen and red flowers with long pollen, we see when the genes are unlinked. When the genes are linked, those aren't here. Those individuals aren't represented in this Punnett square. So linkage affects the types of phenotypes we see when we do crosses. You get much less diversity when genes are linked. Things don't work the way we'd hope. So that leads me to my next question for you, which is something I'd like to discuss briefly, down here at the bottom. given that we know that linkage happens, that sometimes two traits are found caused by genes that are close together on the same chromosome, is it always the case that we can, certainly, combine any two traits we want in any organism we want? And I just showed you an example where when traits were unlinked, we could make red flowers that had long pollen. If that was a desired trait combination we were trying to make, we could do that if the genes were linked. But when they're linked, we set up the cross, same cross, dihybrid self. We expect to see 0 of those individuals. So it's not always possible to make any combination you'd want. So we need to know something about linkage. One of the goals of Mendelian genetics, simple goal. Do memorize this goal. Simple goal of Mendelian genetics. We want to know whether or not genes that cause traits we're interested in are linked or not. It's that simple. Are the genes that control the traits we're interested in combining on separate chromosomes, are they unlinked? Do they independently assort? Do they follow Mendel's second law? That would be good. Or are we dealing with a situation that's a bad situation? The genes that control the traits we want to combine are on the same chromosome, and we expect we might not actually be able to combine those two traits the way we want, just because the genes are linked. They don't independently assort. So as I suggested earlier, at some point some scientists figured out that Mendel lied to us. Independent assortment does not always happen. And the first case of this, again, was Bateson and Punnett, who set up a cross. It's shown here, the one we've been talking about all class, purple and red flowers, long and round pollen. They set up a dihybrid cross, and this is what they saw. They expected to see Mendel's 9:3 to 3:1 ratio. That's the phenotype ratio for independent assortment if genes are on separate chromosomes. They expected to see 9, purple, long, etc. Here's what they actually saw. What sort of a ratio is that? It's pretty close to 11 to 1 to 1 to 3. Does that look like a 9:3 to 3:1 ratio? How close is it to a 9:3 to 3:1 ratio? Not very. Okay, so that's sort of qualitatively, damn it, man, I'm a geneticist. I want quantitative. I don't want very or kinda or sorta. So something we're going to have to do soon is figure out how close to a 9:3 to 3:1 ratio is that. Remember, the goal is to figure out, are these genes linked or not? Are they on the same chromosome or not? If they're on separate chromosomes, if they independently assort, we're going to see 9:3 to 3:1. Those 4 different phenotypes in that ratio. When you see 11 to 1 to 1 to 3, how close is that to 9:3 to 3:1? Does that mean that those genes are linked or unlinked? Looking just at the observed numbers or the ratios here, how has this deviated from the expected ratio? That is, what changed? If you could describe in words, what happened? If it's not a 9:3 to 3:1 ratio, there must be more of somebody and less of somebody else. So what happened? Comparing the observed number of individuals and the expected numbers. We see less of which than we expected to see. Purple, right. So we expected to see 80 purple flower round pollen. Instead, they observed 19. Expected to see 80, observed 27. So we've got fewer of those two traits in the F2 generation than we expected. So who's got more than we expected? The other two types. Purple with long pollen and red with round pollen. look up at this cross. All the pretty colors and pictures. The F2 generation phenotypes, which are the ones that are overrepresented? Which phenotypes do we see more frequently than we expected? Purple with long, red with round. Where do we see those in this cross? These are the parent phenotypes. These are the parental traits, so more vocabulary. So when Bates and Punnett did this cross, they saw more parental types, F2 individuals that looked like their grandparents, the P0 generation, than they expected. They saw fewer than they expected of the recombinant types. Recombinant because these are making new combinations of the parental traits, right? Purple with round and red with long. So the two combinations of traits that weren't evident in the parental generation, those are the recombinant types. Purple with round, red with long, up here. So what they saw was, they saw fewer recombinant types than they expected and more parental types than they expected, and that's how we detect whether or not two traits, two genes, are independently assorting. When we see this shift from the expected numbers, we see something totally different in that observed column. That's our hint. If we see the expected number, if we see a 9:3 to 3:1 ratio, that means independent assortment is working. That means the genes are on separate chromosomes. 9:3 to 3:1. Independent assortment, genes on separate chromosomes. Anything other than 9:3 to 3:1, therefore, gives us the suggestion that we should start looking into the possibility that the genes we're dealing with are on the same chromosome, that they're linked.
Well, can linked genes become unlinked during crossing over?
Yes, exactly. So that's exactly where we're headed. So in the next 2 minutes, I'm going to wind up. But that's where we're headed, absolutely. Is first we start talking about linkage, and then we discover that just because genes are on the same chromosome doesn't necessarily mean that they always go together into the same gamete. Recombination during meiosis, next topic, messes things up a little bit more for us. So I'm going to skip past the next few slides. They were for fun in a sort of nerdy geneticist kind of situation. Maybe I'll talk about them next class. But I would just want to skip ahead now to the last slide, or a few slides. So here's the point, the end of the day. So genes that are on separate chromosomes independently assort. And so genes that are on the same chromosome do not show independent assortment. They don't show 9:3 to 3:1 ratios. So if we can prove that the genes don't independently assort, we have some evidence that they're likely on the same chromosome. That's the goal of Mendelian genetics, to find out if two genes are on the same chromosome, they're linked, or if they're on two different chromosomes, if they independently assort. So next time we start talking about what this picture on the left means, start talking about combination. And what we're really going to start talking about, and this leads into what you need to do for next class, we're going to get back to this question. Are these numbers, the expected and the observed numbers, actually different? Or is it just chance that we observed more parental types and less recombinant types than we expected? So what are we going to do next class if we're talking about comparing observed and expected values? We're going to be talking about the chi-square test. So statistics, I apologize. Next class, we're going to do chi-square test. So this will require you, this was not on the syllabus. There was a third textbook I meant to put on the syllabus. It's free. You download it from the same place you downloaded the first two textbooks. So download this free textbook, read the section on the chi-square test. Okay? Come back to class next time, we'll talk about statistics.